Hello students, this is the lesson video for lesson 4.07 where we're going to be going over genes and alleles. So here's a general overview of what we're going to be talking about today. We're going to be talking about the relationship between genes and alleles, and we'll be defining each one. So genes are segments of DNA that are found on a chromosome. And alleles are variants of those genes or versions of those genes. And because they vary, they have different DNA sequences. So the goal for this lesson is very simple. We just want to make sure that you understand the relationship between a gene and an allele. Okay, first let's go ahead and review what a gene is. So genes are specific segments of DNA on chromosomes. And remember, chromosomes make up DNA plus those proteins. Okay, so genes are specific segments of DNA, and a gene in a body cell usually has at least two different forms or versions, and we call those versions alleles. So there are 23 chromosomes found in all human eggs. Each of those have a unique set of alleles for genes. For example, if we look at chromosome 15 of both an egg and a sperm, there would be genes for hair color. You, your parents, and your friends all have this gene. You all have that hair color gene. However, you all probably have different variations of that gene, and we call those different alleles. So genes, why they are so important is because they really hold those instructions that direct the production of proteins. And proteins are important because they have many different functions or jobs in the body. Basically, proteins are those workers in the cell that get everything done. So by producing various proteins, genes are really directing everything that's happening inside your cells and your body as a whole for growth and development. In the future, we will learn about how a gene's DNA sequence can actually make proteins, but for now you just need to know that that is true. Okay, so we always start with those genes that are in the DNA. In the previous lesson, we learned that there is another type of nucleic acid that helps us out called RNA, but the ultimate product of this whole process is making proteins. Okay, so we use the original instructions in DNA, which are found in the genes, to make proteins. So the genes that govern characteristics such as height, skin, hair, eye color, they produce proteins. Remember, genes produce proteins that play a role in your physical appearance. So variation in those genes, which means actual variation in the DNA sequences, will determine how tall you are and what color your skin is, hair, or eyes are. Okay, so now that we know that genes are specific segments of DNA that code for proteins, now we can look at what exactly an allele is. So an allele is one or more variations of a gene. Okay, so they're all going to be related. All alleles of the same gene will be related to that gene, but they can have different variations. Okay, so if we think of the example of Mendel when we talked about his pea plants, we looked at different characteristics such as flower color. Okay, so you can think of this flower color in terms of a gene. Okay, so the characteristic of flower color is governed by an actual gene. Different colors in flowers, however, are produced by different variants of the gene. So all of the flowers have that same gene, which is flower color, but they may have different variations of that gene. For example, if you have purple flowers, then they must have the purple flower allele or version. If you have white flowers, then they are produced by a different allele of the flower color gene that produces white flowers. Most genes have at least 2 alleles and many have 3 or more. So in humans, many traits also show inheritance patterns that are more complex than just dominant and recessiveness that Mendel showed in pea plants. For example, if we look at eye color, it's really controlled by many different alleles of at least 2 genes. So let's take a look at what that might look like. So the 2 different letters show you that there are 2 different genes that we're talking about, and each of those genes, so the B gene and the G gene, have 2 different alleles, right? We have an uppercase and a lowercase one. And you see that allows us to produce many different types of phenotypes. We have brown eyes, right? We see green eyes, and we also see blue eyes. Okay, so this is an example of how a certain trait such as eye color can involve more than one gene and more than one allele. And so we don't just have the traditional 2 phenotypes or physical appearances. Here in this example, we have even 3. Okay, so we know that each gene has different variations, which we call alleles, and those alleles are going to obviously have different DNA sequences. So that means when we look into the sequence, for the sequences we use those nucleotides adenine, cytosine, guanine, and thymine. They have a different order for those DNA sequences. So if a gene is a specific segment of DNA on a chromosome and alleles are different forms of a gene, then what actually makes these alleles different from each other? So again, the reason why they are different, even though they are usually just slight differences, is that they actually have different DNA sequences. Okay, so the order of the nitrogenous bases, which are these right here, A, C, G, T, really determine what protein will be produced. So different alleles of the gene have small changes in those sequences, which then produces different results. Okay, so different alleles have different DNA sequences, which means we're going to have different results or different phenotypes. So for example, the middle of one allele might contain the sequence CCAT, and let's say that that produces red eyes in a fruit plant, or sorry, a fruit fly, because the allele produces a protein that makes red pigment. Okay, so CCAT produces red eyes. Now, there is another allele for this gene. Instead of having CCAT, let's say that it has the sequence CAAT. Okay, so it has just one nucleotide change, the second nucleotide in the sequence. And just from that one nucleotide change, it results in white eyes instead of red eyes. So you see here, there's a different sequence in the DNA. There's a different sequence in the DNA which produces different results. One red eyes and the other one white eyes. So then this leads us to the conclusion that alleles, DNA sequences, because they change, or sorry, they vary from one version to the next, those sequences must determine the protein that it produces. So if you have a different sequence, it must produce a different protein. Okay, so let's go ahead and look at a real example of a protein called hemoglobin. And hemoglobin is a protein that is produced by a gene that scientists called HBB, and it's located on chromosome 11. So hemoglobin is a large protein made up of 4 chains of amino acids wound together. And its primary job is that it carries oxygen throughout your whole body, so it's pretty important. And the reason why it's able to carry this oxygen throughout the body is because of its shape. Its shape makes it possible for the protein to bind with oxygen molecules and hold onto them as they travel through your body. So geneticists call the allele for a normal adult hemoglobin HbA, or to keep it simple, we're going to say that it's the allele uppercase A, and we're going to call that the healthy allele. Okay, just as a side note, the reason why we call it adult hemoglobin is because there's actually such thing as a fetal hemoglobin that an embryo has when it's developing. Okay, so we know that big A is a healthy allele for hemoglobin, right? But remember, we do have two different alleles for hemoglobin. We have two alleles that cause a development of two slightly different proteins. So we have that one gene, which is HBB, that produces hemoglobin, and we have two alleles. One is healthy, and the other is not. So S produces the abnormal hemoglobin proteins. So allele S produces abnormal hemoglobin molecules which tend to stick to one another and cause red blood cells to become distorted. So they take on a more sickle shape. Okay, so instead of that healthy looking shape like this, we now have blood cells that look more sickled shape. So differences in the DNA sequences of the alleles of A and S account for the different shape. So one really interesting fact is that the alleles A and S are both dominant. Okay, so we call that codominant. So what that means is that if someone was heterozygous, meaning that they had both of these alleles, so their genotype would be something like AS, they would have both healthy and normal, healthy and abnormal hemoglobin cells, and so, or blood cells. And so what that means is that if you have even just one abnormal allele, you still will have that phenotype as well. So you'd have both of them. So individuals that have two S's, which are two abnormal alleles, have something called sickle cell disease. Because they have two S alleles for the trait, those individuals have red blood cells that contain abnormal hemoglobin. So all of their blood cells have that abnormal shape. So the hemoglobin sticks together, giving the red blood cells that contain these molecules a sickle appearance. So the reason why this is dangerous is because it can clot in the body, which causes lots of pain. And can cause anemia. So abnormal adult hemoglobin is produced by that allele S, and the homozygous SS genotype will all have misshapen blood cells. Okay, so if you compare both alleles, so allele A and S, it reveals just one difference between them. There's a switch of a thymine for an adenine in the DNA sequence. So it's just a difference of one nucleotide that causes this condition of sickle cell anemia. And because of that switch, it causes for a different amino acid, which remember, if we have amino acids linked together, right? That's a protein. So what happens is that change in just one nucleotide causes a different amino acid to link in, which causes the hemoglobin to not be normal. Okay, so again, let's revisit the mind map for this lesson. Remember, genes are those specific segments of DNA that are found on a chromosome, and they have those different forms called alleles. Alleles are different versions of the gene, and the reason why they are different is because they have different DNA sequences. And just to take it a little further, remember, if they have different DNA sequences, then they're going to have different proteins. Okay? And in some cases, like we just saw with sickle cell anemia, those different proteins can cause diseases. So here's some important vocabulary. We have gene, which we've seen many times, which is a segment of DNA that produces proteins. We have chromosome, which is a single long molecule of DNA wound around proteins called histones. We have proteins, remember, which are those workers of the cell doing many different jobs. We have alleles, which are a specific form of a gene. Hemoglobin, which is that large molecule that carries oxygen from the lungs and brings it to the rest of the body. And then we have the codominant inheritance, meaning that there's a dominant there are two dominant alleles. So if we have a heterozygous individual, both of the alleles would show up in its physical appearance.